Welcome to Innovation Day 2023, presented by Connect. We've got such an incredible array of things happening here, supporting the life sci and tech communities in San Diego.
Now it is my pleasure to introduce you to our first two speakers. I'm also honored to call them friends. Doctors Kat Jameson and Alysson Muotri, who conduct next-gen cancer and neuroscience research with organoids at the Sanford Stem Cell Institute at UC San Diego. Sharing their amazing bios would take up their entire talk time, so I will not do that, but I'll simply highlight that Alysson is the recipient of the NIH Director's New Innovator Award for his research modeling neurological diseases using brain organoids. He'll talk about what that is. And Kat's research efforts led to the 2019 FDA approval of a new drug for the treatment of myelofibrosis. And I could go on and on. So these are incredible people. And what's really cool is believing sky is not actually the limit. Kat and Alysson will now talk about unlocking the secrets of aging by studying brain and cancer in space in their talk titled Lab to Orbit. So come on up.
All right, I'll get started here. First of all, thanks, Pei-Ling. Thanks, the Innovation Office for the invitation. My pleasure to be here. Pleasure to be with you guys. So you've probably been staring at this wall here, and what the hell, this guy's growing brain in space? And that's a fair question. I mean, why on earth would we go out of the space to actually solve problems here if we cannot do it in here? And there's a reason for that. And one of the main problems that NASA actually face with long missions in space is that the human body is really not adapted to microgravity or space radiation. So that's a very dramatic environment. And one of the things that is most important is that when you come back from these large missions, you come back with serious cognitive decline. So this is out of a manuscript in Science where NASA described the comparison between a genetically identical twins. One stay on Earth, the other one stay at the space station for over a year. And when the one came back, the astronaut came back, there was a serious cognitive decline that persisted after the flight. So why this happened and why this is important? So it's obviously important for NASA because if you're looking for long-term space flights or interplanetary colonization, you need the astronauts to be cognitive sharp. But how to study that? How to know what happens inside the brain? And we have several models. For example, we can take postmortem tissue, but none of them have died yet, so we don't know what's happening in their brains. We study their blood, but blood is really not what happens in the brain. And we have animal models, but the brain size and function and evolution of the mouse brain is very far from humans. So to really understand what's happened inside the astronaut brains, we decided to create or to use a tool from the stem cells where we recreate the human brain in a dish. And this is just like a protocol that we use in the lab. We start from human pluripotent stem cells. These are cells that can make any tissue of the body, including brain cells. And we neuralize them using some extrinsic factors in the tissue culture, and we end up with those spheres that contains about 5 million neurons. These are the size of a bee brain, and you are seeing here is a person in my lab just handling a batch of these organoids. At any time in moment, we have about 50,000 brain organoids derived from live people inside the lab. So what we can do with that, they actually, evolve as the human brain evolves, and they take the same developmental steps as the human brain to a point where they create these neural oscillations. And these neural oscillations is some of these brain waves that you can capture with your EEG. So they actually mimic the same thing. We can even teach them some tricks. So here what you are seeing is a robot learning how to walk using a human brain made in the lab. So it's just taking the plasticity of the human brain to actually coordinate the legs of the robot and using like a feedback loop to actually learn and memorize things. So this is the level of innovation that we are. So if you end up creating something like that, a cyborg in the lab, you end up in the New York Times. And I point to this article because it starts to revealing one of the roadblocks that we might have as we advance, which is the ethical and moral concerns about the entities that we are creating in the lab, something that is not in the philosophy books. I mean, we have to really to relearn how to deal with those things. But going back to space, I mean, my lab has been sending some of these brain organoids to the International Space Station and keep them growing over there for a while, and then we bring them back. So this is all done in a platform that is autonomous, so the astronaut doesn't have to do anything. He just plug this box inside with these human-made brains inside the International Space Station, and they will keep it surviving there for several periods of time. And the interesting part is that when we brought them back, we learned something quite amazing. The brain cells that we have in there has age, and has aged a significant amount. So we can see that by the gene expression profile and the molecular pathways that those neurons now share. They are no longer equivalent of a young, fetal, immature neuron, but they're way more equivalent to a more adult brain. So a little bit of time in space actually age your brain cells quite a while, and that might be the reason why we see a cognitive decline in the astronauts. So we also have been sending brain organoids that are derived from different subtypes of autism. These are neurological conditions that affect not only your cognition, but also the way you behave socially. And I'm particularly focused on a condition called Rett syndrome, because these affect mainly girls, and they end up in a wheelchair. They cannot eat, speak, or do anything alone. So it's a very dramatic, severe neurological condition. So when we send brain organoids from Rett syndrome individuals over there, we reveal something that we never seen here on Earth. We reveal that there is an unleash of endogenous retroviruses inside the cells. Yeah, inside your cells, you have lots of endogenous retroviruses that most of your life are kept silent. But when we put them on microgravity, they unleashed and they start being expressed inside the cells, causing what we call a very dramatic neuroinflammation. This is one of the cytokines that are highly expressed in the brains of people with Rett syndrome. So that was an observation that we never made here. I mean, we know Rett syndrome since the 90s, but we never reveal an inflammatory pathway from Rett syndrome, and not even coming from an endogenous retrovirus. So if now it's an endogenous retrovirus that's causing the inflammation, what if we block the activity of these endogenous retroviruses using drugs that are available, antiretrovirus drugs? So that's exactly what we did in a mouse model for Rett syndrome. And not only we recovered the survival, because they die quite early, but we recovered the cognitive decline that these animals also show. So that's amazing. I mean, we are learning in space some pathways that we could never review here on Earth, and finding drugs that can actually cure or treat a disease. And I'm happy to say that we spin off a company that can take this to a clinical trial. We're starting already next year, recruiting people with Rett syndrome. So fingers crossed, we hope that this medication might at least improve their life quality. But I want to take back to the aging part. And this is our next mission. This is something that we are cooking for a while, which is to send brain organoids from people with Alzheimer's to the International Space Station. And we choose Alzheimer's because it's a very late onset neurodegenerative condition. So it takes years for us to study. The fact that it takes years doesn't allow us to find medications or to screen medications for Alzheimer's. That's why we don't have one. That's why the drugs that we have are not sufficient, because we don't have a good model. We don't have an animal model. We cannot use a human model for that. So that, if I can age the Alzheimer brain cells in space, I might learn something. And more importantly, we are now using a new technology that we are developing. These are microchip, where we can test some of the components coming directly from the Amazon forest. So we are curating some neuroactive molecules directly from the Amazon by talking to the indigenous people that knows about these molecules and testing them for the first time in Alzheimer's organoids that are coming back from the International Space Station. So we are hoping that we will have both applications in space, helping NASA with the astronauts, helping human colonization, perhaps even the tourism that is coming and is becoming hot in the next few years, although it's not yet something significant, but the applications on Earth will definitely be. And these are related to rejuvenation, regeneration, health aging, and we can even use this model to screen drugs here on Earth. So the market just for dementia, it's huge. We're talking about $1 billion market. So any drug, any contribution that you can make to this neurodegenerative disease is going to have a huge impact on our health system all across the world. So I'll finish here by saying that this model can also be applied to other tissues. I mean, I'm looking into the brain, but I can do the same with these stem cells. I can do the same for gut. For liver, for the pancreas. I can understand every single tissue using the same platform. So that's what we are doing now in our labs at UC San Diego. So this accelerated aging can be used to model diseases that takes a long time here on Earth, including Alzheimer's, dementia, Parkinson, and you name it. And most of us, I bet, have someone in our families with one of these conditions. And it's hard, it's difficult. So we hope that this might bring something novel to the plate here. So I'll finish here and hand it to Kat by thanking all the sponsors that we have over the years, and as well as all the family members that contribute to this research by donating their cells so we can create the brain organoid from it, and all members of my lab. Thank you.
It's always very difficult to follow Alysson, but I wanted to thank Payleen and Alysson for inviting me to be here. It's really a joy to know that Houston, we have a problem if you go to the Johnson Space Center like what we've done in the past, but San Diego, we have a solution. So this is why our absolutely innovative culture matters so much. We share technologies, we share ideas, and we do this very early on in the stage of discovery. So I'll talk about precancer stem cell generation and aging stem cell fitness in low Earth orbit, which is what LEO stands for, and talk about this thriving economy that is 250 miles above us. This is something that we're all going to be taking advantage of, and this is something that we've already got lead time bias on. So when I was a kid, my mom would read us the Rubaiyat of Omar Khayyam. He was a philosopher, he was a mathematician, and he was a poet. And he said, when time lets slip a little perfect hour, oh, take it, for it will not come again. So that sense of urgency is something we have in spades here in San Diego. But it's also something I needed on the flight yesterday when I was coming back from a meeting in Heidelberg, because there was a 3-hour medical emergency that I needed to tend to. Unfortunately, we landed. We had to divert to Newfoundland. But this pilot, Kevin, who's the one that let me sit in his seat, I didn't get to take off, but I want. Anyway, he said, this is what teamwork looks like. And this is what we do in this community. We have this strong sense of urgency that Paul insists on at UC San Diego when it comes to innovation. Danielle Lewis, Patrick Chang from our Sanford Stem Cell Institute, T. Denny Sanford, who gave us this extraordinary opportunity to start an institute with 6 centers to go after stem cell aging and pre-cancer. So why space? I think Alysson put this rather aptly. It condenses all the stressors that we have on Earth, but in a compressed time frame. But also we can have the first mover advantage, and that is to look at things that space is unique for in terms of finding solutions. So because of microgravity, we can get homogeneous crystals to do protein crystallography. We can do flow chemistry, and we can also layer down very thin biofilms in a much more predictable way. So I'll talk about the first drug that has ever been informed in its development by space, and that's called rebexinib. After Rebecca Moores, I'll have to tell you about my conflict. I'm the co-founder of Aspera Biomedicines, and we're looking to develop this drug and get into clinical trials next year. So in terms of biomanufacturing, we have improved processes for manufacturing biologics, improved drug stability, and possibly the most important thing for all of us in this room who have made new composition of matter. New composition of matter IP if you make it on a commercial platform in space. So we're going to have 2 commercial platforms, Axiom and Sierra Space, and they will allow us to make new formulations, new drug composition. We can also make stem cell technologies. So this isn't new. Bristol-Myers Squibb has been doing this for protein crystallization together with BMS, or together with Merck rather. Who would have thought that two of the biggest drug companies in the world would partner to do this? And they resolved the crystal structure of Keytruda binding to PD-1, its target. So that's a $20 billion annual revenue-driven product, and they really understood how to use it. Nanoparticle formulation and other things happen in space. So what we did in March of this year is we started the world's first astrobiotechnology hub, and that was to combine our strength in aerospace with our strength in biotech here. So this is the world's first, and we were able to visit Sierra Space to see the Dream Chaser space plane that's going to take off. Guess when? December of this year. And that will take off from a rocket but land on any runway that takes a 737. So that de-risked this environment for drug production. We were invited to go to Axiom Space, as you can see here. And then we went to Houston, and they invited us to go right into mission control, right to the front of mission control, because they know that we've done the most stem cell experiments in space, and we're going to use it as the next thriving economy. And this was all instigated by Alysson, I've got to say. We went there to watch one of his things. So there's Jessica Meyer. She's an astronaut who did her Ph.D. at Scripps Research Oceanography, and there's the crew. So let's get back to stem cell fitness. Have you noticed that some people who are 40 look 80 and other people who are 80 look 40? Our stem cells age at different rates based on stress and inflammation. And what is stem cell fitness? It's about having stem cells that can self-renew, which means to divide without differentiating, become quiescent, undergo differentiation, and live for our lifespans. So this is a journey that stem cells go through, and the essence of stem cell longevity is quiescence. If we lose the quiescent or dormant stem cells, they're not going to last our lifespan, and this is dictated by the niche. So Alysson alluded very eloquently to stem cell aging driven by inflammation, and the bottom line is it looks like microgravity induces this. I'll talk about the blood system, and basically work that I did with Derek Rossi and Irv Weissman. Irv Weissman discovered the cell surface markers for stem cells. Derek Rossi, you all know, is the founder of Moderna. And we talked about the different mutations that occur as our stem cell age continues through our life trajectory and all the issues that arise. And the main question we asked is, why do some people age faster than others and get pre-cancer and cancer stem cell generation and ultimately leukemia? So I'm going to focus on two enzymes, ApoBec and ADAR, and talk about ApoBec3. So the reason ApoBec3 matters, it's primate specific, it's part of our adaptive or innate immune response, and it helps us to overcome retroviral infections, so HIV. It'll be the sushi chef for that and introduce mutations. But it's also the #2 signature in all cancers. So we started studying this together with Lyudmila Alexandrov. We have 7 ApoBec3s. They're human specific. The other enzyme that blocks retroviral integration is ADAR1. That's on our radar because it's active in 20 different cancers as they decide to evade therapy. So we discovered this in leukemia 10 years ago, and then other groups have shown it's really important in cancer. So this is the part where we make a drug to block this. So we decided to make a drug that would stop 20 different cancers from cloning themselves, and this is something that was noticed by somebody in my lab who said, Kat, the splicing modulator blocks ADAR. That's Jessica Pham. I said, I don't think so, Jessica. I think you must have the wrong drug. And of course, she was exactly right. This is a drug we've made with Mike Burkhart in chemistry and biochemistry at UC San Diego. Bottom line is it blocks cancer stem cells from cloning themselves. We've completed the pre-IND studies as Aspro Biomedicines. We're going into completing the IND studies. But for all of you who are developing new therapies, you'll find that the FDA insists that you conform with the Pediatric Research Equity Act and see if it will be useful in children. So we took the FDA's word for it and we tested it in pediatric AML, and lo and behold, this drug really works in preclinical models. So just to sum up, ApoBec is the fuel that gets cancer initiated. This is something we discovered a few years ago with Craig Venter. I saw him walk in here earlier. I know Craig's around there. Craig, he's not listening. Anyway, he's over there. But Craig and I discovered that a few years ago. It's the rocket fuel, but ADAR is the liquid oxygen that allows cancer to blast off. So just to let you know why the NASA twin study was so informative, you get one guy to go into space, and that's Scott Kelly. He comes back 2 inches taller than his identical twin brother, but he has these pre-malignant changes that include inversions and translocations in his chromosomes, and then inflammatory growth factor upregulation that leads to both APOBEC and ADAR activation. So what we did is we decided to write a grant to NASA, this is Alysson and I and others, to look at could we use space as the ultimate stressor to see could we study accelerated aging but in a compressed time frame in the brain and the organoids that Alysson developed, in the bone marrow and nanobioreactors that we've developed, and in the liver. And so we got this rather large grant from NASA, which was covered in the Washington Post, created a little bit of performance anxiety, but we've done the most stem cell experiments as the iScore team, Integrated Space Stem Cell Orbital Research Center, that Alysson is the director of and I'm the co-director of, in the world and out of this world, actually. So what we decided to do is take 8 normal individuals, put their stem cells in a nanobioreactor we created, and look at ground controls. And what we found was hyperproliferation in space. And this is the nanobioreactor going to the Cube Lab. This is the autonomous system that we developed with Space Tango. And these are the cells. So what happens in space when we use this fluorescence ubiquitination cell cycle indicator that we developed with Roger Chen's lab, you remember he got the Nobel Prize for green fluorescent protein, is we see the maintenance of these red dormant stem cells on the ground after about 21 days. But in space, we get this proliferative burst and we start to lose that red dormant stem cell population. Together with Ludmila Alexandrov, we learned how to measure telomere length, and we found the telomeres shortened in space. And then we also found that the stem cells didn't work properly. They didn't make colonies. Those colonies didn't clone themselves. They didn't replate. And then this goes back to the APOBEC story. In space, they activated that enzyme, APOBEC3. This leads to pre-leukemic changes that look exactly the same as if we overexpress APOBEC. And this is the piece of data I was alluding to that Craig and I got together. If you look at patients with myeloproliferative neoplasms, they have the same APOBEC signature as people who have gone into space for just 30 days. So what we're looking at is the same thing as Alysson, the repetitive elements or human endogenous retroviral sequences. It looks like there's a component that get activated in space within just 30 days. And there's one in particular that portends a poor prognosis in AML. But line element expression goes down, as you would expect if APOBEC goes up. So we're seeing a reduction in ADAR, increase in APOBEC, so advanced aging. And then in the last couple of slides, I'll just show you. So if we find that normal stem cells get exhausted in space, what happens to cancer? So we made these tumor organoid models to look at cancer on the ground and in space. And what we found was in the colorectal cancer model that you see there, cancer tripled in size in space in just 10 days. And it activated that cloning gene called ADAR. So in the second mission, the Axiom-2 mission, we decided to test, can we block ADAR with this green fluorescent protein reporter reading out ADAR in a tumor organoid model. This is triple negative breast cancer. You can see very bright GFP expression. We add fedratinib, which is that drug that Palin was saying I was involved in co-developing, which is correct with the IMPACT team and Targogen. And then it didn't quite dampen ADAR, but rebexinib did in a very specific and powerful way. So now what we're studying is the AX2 crew at single cell sequencing depth, whole transcriptome, whole genome, to see do they age faster, to see can we predict and prevent this accelerated stem cell aging and precancer development by using space to do that. So that's where we're at. I'd really like to thank you for having this opportunity to be able to present our work here. And then just to say the most important thing about all of this is how we mentor each other. Plato said this in the Timaeus, but to mentor the next generation of scientists is just a treat. So thanks everyone. I would like to thank the whole team and you for listening.
I don't know if we have questions.
Thank you, guys. That was fascinating. I've heard it before, but every time I learn something new. Thank you so much. I actually have a bunch of questions, but first I would like to ask to keep the conversations lower. This room, unfortunately, has terrible acoustics. It becomes very difficult to hear our amazing speakers. They've taken the time to come here, and a lot of people want to listen. So please, 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 keep the conversations down. So appreciate that. With that, we have a couple of minutes for questions. I have some, but I will give it to you guys first. So anyone? Any takers? Sky's not the limit, and these guys know it, so here you go.
I believe we just had a record-setting astronaut come back from the space station. Are there plans to study what happened? I think it was, 2 years or something, or 3 years? Just be curious whether that fits into your research.
Yeah, so I'll comment first because we're meeting with the team from EPIC. So as you know, EPIC was founded by Judy Faulkner. She's a 7.1 billionaire according to Forbes. She made this system that has 150 million patient records in the US, so it's the biggest electronic medical record. I suggested that we have an astronaut biorepository for data on EPIC, and it would be a patient like me portal, but this is an astronaut like me portal, so that they will have their genomics data integrated into EPIC, and then they'll be able to look at what do other astronauts like them look at. So there are only 600 people that have been to the International Space Station, but as Alysson alluded to, this is going to be a lot more. So we're meeting with the EPIC team on September 25th to talk about how to do that. NASA wants us to potentially host the biorepository and the data here because we have the San Diego Supercomputer Center. So we'll be able to have a fully integrated data set. And then Alysson, of course, can look at all the brain organoids that derive from blood. Do you want to comment, Alysson?
I know, I think Kat says it all. I mean, having the biorepository will allow us to make comparison and understand if there are people that are more resistant, less resistant, and find ways to protect the astronauts.
Any other questions? Well, I have one, and that's really about how do you then create the control on Earth? Can you give us an explanation of what happens in space and what happens on Earth, assumed synchronously, so that we understand the delta?
Yeah, I can start. I mean, we usually have both asynchronous and synchronous control, meaning the same experiment that goes over there, we replicate on Earth, and we follow the two of them in parallel. Sometimes things doesn't go as expected, so we have to make like small adjustment. That's what we call the asynchronous control. It's another set of experiments where we adjust along the way to mimic the best conditions in space.
Yeah, we do the same thing with the nanobioreactors we created where we can study stem cells from the bone marrow long term. We do that on the ground and in flight. So we have an exact same replicate on the ground and in flight. And we get real-time images. You know, we're getting images from the automated bioreactor from space and on the ground, but also when we have a crew-tended mission like we did with Peggy, we do this with crew, putting our bioreactors on the microscope, and those are the images I showed you there. And that's the first time cancer has ever been imaged in space, human cancer, and we got real-time images with a reporter.
Amazing. Thanks so much.
I really feel passionate about sharing the San Diego talent, elevating San Diego, because it is the best place to be an entrepreneur.